Hi, welcome to PK's classes. Today we will continue the nervous system with the main cell of the nervous system, neuron. And this neuron or nerve cell has a cell body, okay? And the cell body is having different processes, okay? So in this diagram we can say that 1 2 3 4 5 6, okay, 6 processes are there, and one of the process is called axon and the other processes are called dendrite, okay? So in this dendrite we can say like this. Different subdivided also the process, and cell body has a nucleus, and this axon is having the axon terminals, okay? And these axon terminals, at the end we have synaptic end valve, okay? And like this we can have. So neuron, a cell body is there, then axon and axon terminals are there. So we can divide a neuron into 3 parts: cell body, okay, cell body, axon and axon terminals, and the dendrites, okay? Three parts: dendrites, cell body, axon and axon terminals. Now the supporting cell, we know the supporting cell is called neuroglia, and this neuroglia, one of the neuroglia surrounds this axon, okay? So if we take a transverse section of this one, this will be like this, and so this is the supporting cell. This can be a swan cell in ANS, and this can be oligodendrocytes in CNS. Okay, we'll discuss this neuroglia in detail later. And this one is called myelin sheath. And this myelin sheath surrounds this axon, okay? So this neuron can be divided into two types: myelinated and non-myelinated. This is myelinated one, and this open spaces is called node of Ranvier. So when impulse goes, it jumps from one node to another node, because this myelin sheath makes it insulated. So the impulse jumps, whereas in non-myelinated cases, the impulse, we can say walk, okay? Impulse goes along the axon, whereas in this case it jumps. So impulse conduction is faster in myelinated. Whereas in impulse conduction is slower in non-myelinated, okay? So if we see this diagram, we can have this one. This one is myelinated, okay, we have discussed, and these are the axon terminals, and these are the synaptic end valves, okay? And this is a case of non-myelinated non-myelinated, and there is no myelin sheath. Here myelin sheath is present. This is the difference, myelinated and non-myelinated. And if we take a whole picture of this synaptic end ball, how it looks, it will be like this. It will be like this, and within this there are vesicles. There are vesicles. These are called synaptic vesicles. Synaptic vesicle. And inside the synaptic vesicle there are chemicals stored, and these chemicals are called neurotransmitters. Why they are called neurotransmitters? Because they transmit impulse from this nerve ending to another one. Suppose another neuron is there, that means from this nerve ending to another neuron impulse can go. So this area is called synapse. Okay. On the basis of this, these are called synaptic vesicles. Synapse, also called synaptic cleft. Okay. And the neurotransmitters, when the synaptic vesicle membrane is fused with the axon terminal membrane, the neurotransmitters are released into the synaptic cleft, and then these neurotransmitters can act on their targets in the presynapse as well as on the postsynapse. So this is called presynapse, and this is called postsynapse. So if a receptor is there, this is called presynaptic receptor, and if the receptor is present on the postsynapse, this is called postsynaptic receptor. And these neurotransmitters can act on the presynaptic receptor or postsynaptic receptor to show their actions. And also these neurotransmitters are degraded by enzymes in these synapse or synaptic cleft. So if this is a neuron, then this is called synapse or synaptic cleft, and if this is a muscle, suppose this postsynapse is a muscle, then this is called neuromuscular junction. Same thing, this is a neuron, this is a muscle, so this junction will be neuromuscular junction. Also, if this is a gland, then this is called neuroglandular junction. Okay, neuroglandular junction. And if this is a, commonly we can call it neuroeffector junction. So this effector may be neuroeffector junction. This effector may be a neuron or a muscle or a gland, like that. Okay. Now, these neurons can also be classified as multipolar, bipolar, and unipolar. So unipolar means the process is one process, so that means cell body, only cell body, and one axon is there. Okay, we can draw a simple diagram: cell body, axon, axon terminal. So if this is this is called unipolar, and in mostly sensory neurons. Okay, so again, neuron can also be sensory or afferent and efferent or motor. That means the neurons coming from sense organ to CNS, sensory or afferent, this way, and from CNS to periphery, that is called motor or efferent. Okay, and most sensory neurons are unipolar. Okay, and most neurons in brain and spinal cord are actually multipolar. Okay, that means dendrites are there also and axon is there, and few are bipolar. That means one axon, one dendrite. So that is bipolar. And for example, retina, eye, inner ear, olfactory area of brain, all these contain bipolar neurons. Okay, and this myelin sheath provides the white color to the area of brain and spinal cord. So if we see in the brain and spinal cord, suppose in brain, if you take a transverse section, so this part, the central part will be white and the peripheral part is grey. So in brain, that means the myelinated neurons are located on the center of brain, so that provides the white color, whereas the rest part is grey color. Grey, this is white. And just the opposite in case of spinal cord. This is in brain, and in spinal cord, just the opposite. Okay. And the periphery is white because of presence of myelinated neurons, and the central part is the grey because of presence of non-myelinated or the presence of cell bodies. Okay, axon terminals, mainly the axons, myelinated axons or neurons, nerve fibers are tracts are present in the peripheral part of spinal cord and central part of. Okay, in the next class we will study the neuroglia.